Hello. So I just would like to introduce Nicole Krentz, who's from Stanford School of Medicine, and she's going to talk to us a bit about the work that she and her colleagues have been doing on the REB1 gene and how it's linked to diabetes. So, Nicole, could you give us, just before we start, a little bit of the background to your field of research and explain some of the fundamentals about what islets and beta cells are and how they're involved in diabetes, please?
Absolutely, yeah. So islets are clusters of cells that are found in your pancreas, and they really help to control your blood sugar levels. So within the islets are the pancreatic beta cells. These are the ones that make and secrete insulin, and that's a really critical hormone for your glucose homeostasis. So when you eat a meal and your blood sugar rises, the beta cells can sense this, and they secrete insulin, which then acts on your peripheral tissues, things like muscle and liver, and tells them to take up that excess sugar and store that energy until your body needs it. So what happens in diabetes is that the beta cells don't make enough insulin. And so you end up with too much sugar in your blood or hyperglycemia. And in type 1, this is due to an autoimmune destruction of your beta cells, whereas in type 2, which is the most common form of diabetes, this is a combination of genetic and environmental factors, and it results in your beta cells no longer being able to make enough insulin, and you end up with that elevated sugar in your blood or that hyperglycemia.
And then, so you part of your study is looking rather at the genetic side of what might be causing these problems with glucose metabolism. So you've been looking at a gene called REB1. Well, what was already known about REB1 before you started working on it, and what made you so interested in it?
Yeah, absolutely. So our lab, we're really interested in understanding how diabetes occurs. And as you pointed out, this is taking a genetic first approach. And what we're really interested in, why do those beta cells fail? And so we use human genetics, in particular genome-wide association studies, and that identifies regions of the genomes or variants that are associated with altering your risk of diabetes. And so our team and others had identified genetic signals at the REB1 locus, and those were associated with both type 2 diabetes and other glycemic traits, including fasting glucose. And so this suggested to us that the REB1 protein is likely involved in glucose regulation and therefore in diabetes. And so what we knew about REB1 at the time was that it's a transcription factor, it's broadly expressed, and it had previously been implicated in cancer, as well as a rare syndrome in humans called Noonan syndrome. But what REB1 was doing in diabetes or in the islet itself was previously unknown.
So that's interesting to be able to carry out this groundbreaking research on REB1 and diabetes. So what kind of models did you use to actually conduct your study?
That's a great question. We're particularly proud of this paper because it's an example of how collaborative science is really key when you're trying to dissect these disease mechanisms at genetic signals. And so we collaborated with several groups, including Dr. Marcel Den Hood. His team is at Uppsala University in Sweden, and they generated a zebrafish model that was lacking the orthologs of the human REB1 gene. And then in Oxford first, and now at Stanford, myself and Dr. Katia Mattis, who are part of Dr. Anna Goin's team, we worked and used several human cellular models, in particular of the beta cell. So we were trying to understand what REB1 is doing in human beta cells. So this included the human endo-C beta H1 cell line, which is a human beta cell line, as well as human-induced pluripotent stem cell-derived beta-like cells. And then finally, we wanted to bring it back to the humans and ask whether carriers of REB1 alleles have altered islet cell function. And so to do that, we collaborated with Dr. Patrick McDonald at the University of Alberta in Canada.
And so that was looking at then islets. So looking through these several models, did you see some similar things between the models? Were those the kinds of findings that you found?
Yeah, absolutely. So there was really a few key things that came strongly across throughout the different models. So we found that REB1 is required for normal insulin expression and for insulin secretion under prolonged stress. And interestingly, we found that human carriers of several REB1 alleles, they actually have altered islet cell function. So this suggests that the genetic association of REB1 with both type 2 diabetes and other glycemic traits might actually be due to how the cells develop and function. So REB1 is required for normal islet cell development and function. And when it's perturbed, you end up with dysfunctional islet cells.
And so you could see this both in the human cell lines and also in the donor islets.
Absolutely, yeah.
Oh, very interesting. So do you have an idea of what the mechanisms could be behind these changes that you could see with REB1 expression?
That's always the toughest question. I think our study definitely supports a key role for REB1 within the islet itself. We think that this is likely due to REB1 transcriptionally regulating genes that are important for both how islet cells develop and how they function. And so when the expression or function of REB1 is disturbed, as in, carriers of human REB1 alleles, your beta cells do not develop properly and they don't function as well and they produce less insulin. And that predisposes you likely to diabetes.
So it's almost as if REB1 might be the master switch that kind of affects several other genes that are also involved, and some of which perhaps are known and some of which may be less well known.
Absolutely.
Yeah. So what's the next stage? What do you hope to do next in terms of your research?
Yeah, in this study, we identified that REB1 is really key in beta cell development and function. But I mentioned earlier that REB1 is expressed in multiple tissues, including other diabetes-relevant tissues. So we're currently following up on REB1 is doing what it's doing outside of the islet, particularly in adipose tissue, and how this might contribute to changes in diabetes risk. And excitingly, I will actually be following up on some of these remaining questions in my own lab, where I plan to explore the role of REB1 and transcription factors in cell fate.
Well, that's exciting. Yes. Well, we wish you the very best of luck with your further research.
Thank you so much.